So let me introduce you the second speaker. So our second speaker is Will from Glasgow, for the CVR in Glasgow. So he's going to talk about phenotypic evolution of SARS-CoV spike during the pandemic. So Will has been working a lot on the molecular determinants of SARS-CoV, so how this determine cross-species barrier, transmission, and viral pathogenesis. And he has also been involved in study the different SARS-CoV-2 variants, their fitness and the virulence. And the main technique using is cellular biology, and what they're really good at CVR is reverse genetics. So I guess I'm sure he's an expert on that too. And now Will is focusing on understanding experimentally how coronaviruses evolve and what phenotypic traits are important and determine viral fitness, how these viruses interact with the host in the human, especially in the human respiratory tract. So I hope it's more or less what you do, and I let you talk for the next half an hour.
Hi everyone. Thanks Julia for the very nice introduction. So let's start straight away. So I think it's now 5 years later after the beginning of the pandemic, or a little bit more than 5 years, and I think it's always good to remind people exactly what this pandemic has caused. Also, I'm sure that this audience doesn't need to be reminded, but as of last month, from the WHO data, the COVID-19 pandemic has caused over 7 million deaths, and obviously after billions of infection. And we all know that these figures are actually underestimated. So basically, a completely new virus emerged in the human population a bit more than 5 years ago and has caused the death of many million people, and maybe even more since because of the long COVID, etc. So this is what the COVID-19 pandemic instead looks like from a computational point of view or genomic point of view, with basically all these different variants, because the virus keeps evolving over time and leading to the emergence of different variants all over the place in the world, and some of them being fit enough to basically replace, so each new variant replacing the previous one and then causing different waves, as shown in this plot here, that again all this audience is probably aware about. And then it is known that most of the variants defining mutations are actually highly concentrated in spike. So obviously, all over the genome, there are mutations popping up, but it turns out that most of them are concentrated in spike. And this is due because probably this is where the pressure of selection is acting the most. And I'm not going to go into details about this because the antigenicity in immune evasion related to spike has been extensively studied, and that's not exactly what we are doing in this group of G2P and at CVR. We are more interested on the other aspect, like the pathogenesis, the fitness of the virus, and potentially linked with interaction with the host. And basically here at CVR, along other groups, we could show that for example, from the emergence of Omicron, there was a quite drastic change in the way that the virus was entering the cells, and that was then shown by other groups. This was down to spike, and that was also at the same time affecting the pathogenesis and or the virulence of the virus because of the spike that has changed. So here we really wanted to step back a bit and say, okay, so some variants were quite of major and were already characterized, like Alpha, Delta, then Omicron. But then we really wanted to look at what could have happened before the emergence of Omicron by looking at other pre-Omicron variants, and especially their spike, and then also move towards the Omicron era, and then investigate in the post-Omicron era how the spike has changed different phenotypes, to basically look at the evolutionary trajectories of SARS-CoV-2 spike and the associated phenotypes during the pandemic, and this by using a genotype to phenotype approach. So to achieve that, we basically selected a panel of different variants based on the location they did emerge in the past, so basically all over the place. And also the plan was to basically span from very early on in the pandemic with the D614G, which was basically the first variant, let's put it that way, and then spanning all along the pandemic until BA.286, which is now a bit more than a year old. Again, we could have kept going like this for months and years, but we had to also draw a line and stop this study. Otherwise, it's a never-ending story. And the plan was basically to use the reverse genetic system that was established here at CVR to basically create or generate this library of recombinant viruses. So basically, we took the genome, the wild-type genome, and then we swapped the spike gene from many different variants, and we generated 27 of them. And the plan was to characterize this library and assess this library in terms of phenotypic traits using in vitro, in vivo, ex vivo, and a model for this. So basically ranging from a very classic growth curve in Cal3 cells, which are the gold standard for coronavirus or SARS-CoV-2, I would say. Then also looking at the cell-cell fusion or the capacity of spike to mediate cell-cell synthesia, and then also look at the protease usage, assess the virulence and the pathogenesis in hamster model, and also focus a bit more on what's happening in reconstituted human airway epithelium. So first, to try to give a brief overview without overloading with data, we first, so like I said, characterized these viruses in Cal3 cells, and where we could basically see that within the pre-Omicron era, they didn't change that much. Of course, some did perform a bit better in the cells like the Lambda variants or the Delta variants, but nothing really dramatic. Then from the emergence of Omicron, the first Omicron, we could basically observe this drastic and significant reduction in the replicative fitness in lung cells, which was sort of maintained in next Omicrons until the most recent ones, like XBB.1.16, EG5.1, or BA.286, which basically did recover the capacity to infect and replicate fine in lung cells, so to the same level as wild type, or actually even better. At the same time, like as mentioned in the introduction, we also wanted to check what kind of physiogenicity is associated with those spikes and basically how it is affecting the formation of synthesia. So using a split GFP system, which basically will have a cell population that express, let's say 90% of the GFP protein that doesn't emit any signal, and then you have another cell population that just encodes for the last bit of GFP. Again, not emitting any signal. And then when you co-culture these cells and infect with the virus, you can basically, upon the formation of synthesia, have the GFP being recovered and so with signal being emitted. And we did measure this over time, and this is what basically it looks like, but maybe a way that is slightly more easy to digest by using this heat map. We could see that in the pre-Omicron era, they basically didn't change that much in the way they were causing stem cell fusion. But in the Omicron and post-Omicron era, we could see that, again, that was associated with a drastic drop in the capacity to mediate synthesia formation. But again, over time and towards the most recent Omicron variants, this capacity was restored. Okay, so this is all nice and cute, but this is all in vitro. So we really wanted to assess, okay, spike is changing many different things and different phenotypes, but is it actually causing differences in virulence or pathogenesis in the animal hamster model? And so we assessed a subset of this library of viruses. And here, focusing first on the pre-Omicron variants, we could see that the virulence in terms of weight loss here did not change or did not vary that much between these different variants. While for the Omicron and post-Omicron, we could see that first like the BA1 spike or BA275 were associated with a much milder and attenuated virulence in hamster model, while some others actually managed to restore or be a bit more like the wild type virulence. So suggesting that, again, this is very dynamic, and depending on the mutations carried by spike, this is affecting the virulence of the or the pathogenesis caused by the virus. And this was also confirmed because weight loss can be influenced by many different factors, including just the behavior of the animals. We also developed here at CVR the digital pathology pipeline, by Vanessa Herder and some other colleagues, where basically we could analyze the whole lungs of these infected animals, looking at different markers like IBA1, which is for the macrophages infiltration, and CD3, which is for T-cell infiltration, so that would be down to the inflammation. And we also looked at the TTF1 marker, which will be looking at the proliferation of the type 2 pneumocytes, which basically will take the space upon the appearance of the lesions. And basically, we could see by quantifying this using AI-assisted software that, again, in the pre-Omicron era, that didn't vary that much in terms of virulence and marker of virulence. However, from Omicron, we could see that the first Omicron was associated with indeed an attenuated phenotype, which was also shared with some other, but also other variants such as BA5, EG5 were actually kind of the way upwards to more restored virulence compared to wild type. So I also mentioned in the introduction that we really wanted to focus a bit more on the human airway, and especially in the upper airway, because this is actually very critical since this is basically where the virus was first be in contact with the host, and that will need to be efficient to actually establish a successful productive infection to replicate in the host, and then later on potentially be transmitted. And we thought that this was actually slightly neglected or actually not investigated enough. So for that, we use reconstituted human nasal epithelium, where basically you can observe using air-liquid interface cultures, you can recapitulate the human epithelium in the nasal cavity with having a mixed cell population at the air-liquid interface. And this is a very good model, and I think a very good proxy to look at those questions. And indeed, when Omicron emerged, we here at CBR, but also other groups in the world, did show that actually in those cultures, Omicron was having an advantage over Delta, which was basically the variants it was replacing at the time. And we really wanted to assess if this was really down to spike. By the time we were doing this study, some other studies came out showing that indeed it was spike, but they only stopped at the BA1 spike. So we really wanted to investigate a bit further and explore a bit more. And basically, the take-home message from there is that you can see that all the pre-Omicron spike variants are grouping together here with the wild-type spike, while all the post-Omicron spikes in those recombinant viruses are actually associated with a greater replicative fitness in those cultures. Interestingly, I'm just going to stop here a little bit and draw your attention of one variant, which is called A30, which is a pre-Omicron variant. It's actually a tiny lineage that was that reached Africa and started to spread a little bit there, but then stopped very rapidly early on in the pandemic. But at the time, it had quite a high mutation profile, and we, like I said, we also did look at the protease usage, like temprases 2 and cathepsins by the virus and by this library of viruses. And here, just to show a subset, which show that wild type was basically highly inhibited by camostat, so meaning that it's highly dependent on temprases 2 for entry, the same as Delta, while Omicron was basically switched and basically relying a bit more on the endosomal pathway. And actually, we observed that for A30, that was showing a similar phenotype, so suggesting that A30 was also preferentially using the endosomal pathway. So we all thought that basically this switch only happened first in Omicron, but it actually already happened before. And also, we all thought, even including us at CVR, that this was actually linked with the replicative advantage in airway cultures. But it turned out that to not be the case, because otherwise A30 will be grouping here with the Omicron recombinant viruses. So it seems that this threat is actually a little bit more complex than what we initially thought. And then we really wanted to take advantage of the tools that we established here, especially from the animal work side. And basically, we really wanted to look at those cultures and basically what's happening in those cultures, because we also said that the virus, the Omicron viruses are replicating better, but how does that affect those cultures? And here, by staining for the viral spike RNA by in situ hybridization, we could see that first, all these cultures are highly positive. But what you could see here is that all the cultures infected with the post-Omicron recombinant viruses were associated with this crap detaching from those cultures. And then this crap actually has an actual term, which is called slough off. So basically, because of this efficient entry and replication into cultures, this is causing the cells to detach and basically to slough off, and they are full of virus. So this could potentially have a role in the transmission later on. And we did quantify this accurately using AI-assisted software, and we could indeed confirm that this was higher, or this phenomenon was happening more in the Omicron-infected cultures compared to pre-Omicron. At the same time, we also wanted to look a bit at the interferon response, because this is very important in the way the virus will be controlled or not. And here, by staining for IFIT1, which is an interferon stimulated gene, in those same cultures, we could see that all the Omicron infectors were highly positive for this IgG, so suggesting that it's triggering a lot of antiviral state. And this was also confirmed by Western blot, where we could see that the post-Omicron infected cultures at 24 hours and 48 were already producing more, having more phosphorylated STAT1, suggesting there were more interferon being produced, and then they started to respond with indeed an upregulation of IgGs like IFIT1 and viperin, which was higher compared to pre-Omicron before catching up at 72 hours. So it seems that these cultures, the virus is replicating fine, despite triggering a lot of interferon response. And a very nice paper from Alex Compton's lab in the US came out a year ago, showing that indeed spikes might help to overcome the interferon response in those cultures, where basically they did pre-treat those cultures with interferon beta or interferon lambda, and why they could observe a significant reduction for the wild-type strain or the wild-type encoding for the delta spike, so a kind of recombinant virus. The post-Omicron one were basically not affected by the interferon pretreatment. And this is something we were actually looking into a little bit, not into much details, but basically we could also take another approach by, instead of pretreating the cells by interferon, we actually treated with RUX, which is ruxolitinib, which is an inhibitor of the interferon response, let's put it that way. And why we could see that indeed Omicron, or an Omicron spike was replicating better than the pre-Omicron. Once you pre-treat with RUX to shut down the interferon response, those pre-Omicron are actually capable to replicate much better. But we could see that for the Omicron spike, that didn't really change that much. So indeed confirming that regardless if you block the interferon response or not, the spike was already giving an advantage to the virus and basically overcoming the interferon response, which somehow links with a study that we did contribute by collaborating with Claire Joly and Greg Torward's lab down at UCL in London, where basically they did show that even in the Omicron era, the virus was first very bad at antagonizing the interferon response, as you can see here with BA1, BA2 triggering a lot on interferon beta and ISGs. Why then, from BA5 onward, there was actually a better innate immune antagonism. And if you try to combine a little bit all together, it seems that, or at least I would think that SARS-CoV-2 spike could drive selection for better innate immune antagonism by non-spike genes, because as a better and more efficient replication in those airway cultures trigger a lot of interferon response, that's actually pushing the virus to select for better antagonism. And this is the kind of model we can draw from this, that basically Delta compared to Omicron, or let's put it the other way, Omicron compared to Delta will infect much more cells early on and in the first stage, and then obviously produce more progenies that then would spread faster and better compared to Omicron. This is at the same time causing a lot of different response, which the virus didn't seem to bother. And then also causing, as this massive replication, as a consequence of this massive replication, the cells detaching at 4 days post-infection. But interestingly, I didn't show it because in interest of time, we also extended this experiment up to 7 days post-infection, and actually we could see that the pre-Omicron, like Delta spike, could actually also trigger this slough off. So it's just basically the kinetics of these key events that is changed because of spike in the upper airway. And then we also wanted to check if these features, these traits, was maintained in subsequent Omicron variants. And indeed, when you look at all these spikes, they all maintain this greater replicative fitness in those cultures. And more interestingly, they were all the same until BA286 spikes, which did show another level of advantage over the BA2, which was the parental strain at the time of variant. And basically, now we all know that BA286 was the seeder of JN1, which took over after also acquiring mutations to evade antibody immunity. Okay, so we observed this phenotype that clearly first appeared in Omicron and then got maintained, but we don't really know what are the molecular determinants in spike of this enhanced nasal tropism or enhanced fitness in those cultures. So basically, in other words, what makes Omicron Omicron? So Omicron actually has got a unique set of mutations, which basically only appeared first in Omicron and then got maintained in every single one of them. And basically, these are 13 of them. So here, just a cartoon illustrating our approach here, because we really wanted to take advantage of our reverse genetic systems to assess this a bit more into details. So basically, the approach was to take the wild type spike, okay, and then introduce either all together, like here, these 13 unique Omicron mutations, or then in different clusters, so just like these 4 mutations found in the RBD, or these 4 in the RBM more specifically, or then the other 5 that are basically shared between the furin cleavage site and the S2 domain, and to assess these viruses in nasal cultures. Here, the take-home message is basically, doesn't matter if you introduce this mutation by cluster or all together, they do not confer this replicative advantage in airway culture. So we do not recapitulate the phenotype we observe with the full Omicron BA1 spike, which was somehow a bit disappointing, but well, it is what it is. Then we also wanted to take the approach from another, so the other way around, basically starting from the Omicron BA1 spike that we know has or does confer the phenotype, and then revert either altogether these residues back to wild type or again in clusters. And here, what was really striking is that, first of all, from the moment you start reverting some clusters or altogether, you kind of really lose drastically this replicative fitness in airway cultures compared to the Omicron controlled spike. But more interestingly, we could see that this mutant, which is basically only looking at the at the mutations that are found in the RBD, when you revert these guys back to wild type, you completely kill the virus in those cultures. So we thought, okay, it's maybe just because the overall fitness of the virus is not good enough. Basically, it's a crappy virus. We already knew that from the rescue, it was actually clearly fine. But then we assessed the replication of this virus in carrier-3 cells and could observe that this virus that was completely dead in nasal cells is actually perfectly happy in carrier-3. It's actually restoring the level of wild type, so being better than the Omicron spike. So there is clearly something going on here. What exactly, we don't know yet. That's something we are really willing to follow up. And basically, these 4 mutations are localized in the RBD here, and that's where they are localized here. And using AI like AlphaFold, protein language model, etc., here at CVR, these are very preliminary data, but basically we could see that these mutations that are part of the RBD, so 371, 373, 375, are actually having the greatest influence on other mutations around. So just suggesting that when you revert them, there is some sort of, structural problem, or it's not very stable anymore, to basically affecting the potential phenotype. But that's something that we are trying to follow up. And basically, the take-home message is that given mutations might only have an effect when they are in the given structural context or genomic context. So this is a clear example of epistasis, but it is very complex to address. So to summarize, I hope I convinced you that spike is not just about antigenicity or immune evasion. It's actually, antigenicity is very important, but also as important as the actual virus fitness in the human airway, because it's all playing around, and these are the main drivers of transmission. And then all the different phenotypic traits associated with this are maybe more like byproducts. So they are also affected when you are changing the mutation or changing the residue in spike, but they are just consequences of what has been selected for early on for transmission. And indeed, we could show that by changing spike, and so by having different mutations in spike, this is drastically changing the way the virus will replicate in the upper airway, but also the way the virus will infect and replicate in the lung, which then will affect the virulence, the pathogenesis. So I think overall, this clearly shows that spike first is a key determinant of transmission and virulence, or replicative fitness and virulence, which is under constant adaptation to human. And I think it sounds very trivial what I'm going to say, but I don't see it a lot in the literature. I think it's time to accept that we as human are the new reservoir, or are the current reservoir of this virus that has gone through a lot of adaptation. And then I just would like to use the last few minutes, if I may, to try to open a bit more here. And I think with the previous presentation from Rachel, that really fits in there. So we observed that during the pandemic and all these different variants, because of the evolution of the virus, that was associated with drastic phenotypic changes during the pandemic. But this is just SARS-CoV-2, which is basically fitting here in the sarbecovirus, so the SARS-related beta coronaviruses phylogenetic tree. So all these guys are actually there, but they are similar in some ways, but they're also pretty distant. Let's not forget that between SARS-1, the original sort, and SARS-CoV-2, there is only about 80% identity, and at the spike protein in terms of amino acid, it's only 76% identity or 86% similarity. So it's close, but it's actually pretty distant. And we could see that they behave completely differently. So we should not assume that all the sarbecoviruses will behave the same. I don't think that will be the case. And indeed, if we then explore a bit more and put this more into the context of the large diversity of coronaviruses, this is actually difficult to assume that they will all behave the same, or one group of viruses will all behave the same, because if we look at the recent literature, we all thought that, for example, MERS-related coronaviruses, MERS-becoviruses, because MERS is using DPP4 as a receptor, they all assume that, well, they all use DPP4. Well, all these recent papers are showing the opposite, that actually a lot of them, or actually the ones that were tested so far, could actually use ACE2, either human ACE2 or their bat species ACE2. And I think this is kind of maybe talking about some sort of convergent evolution, or maybe the ACE2 receptor usage was maybe the ancestral traits that then got explored and changed over time. So some of you might know that Massimo Palmarini, which I work with here at CBR, is moving to the Erasmus Medical Center in Rotterdam to take the directorship over there, and I will be also moving over there and basically try to really strengthen the collaboration between CBR and Erasmus to really investigate the zoonotic potential and evolutionary trajectory of these different coronaviruses from genotype to phenotype. So with that, I just would like to end here and thank everyone involved on this project. It's really a team effort. I'm basically here only the messenger today because, for example, all the reverse genetics, the design of the constructed thing is all down to Vanessa Cotton here. And then with Judy, we did actually all the characterization in vitro and ex vivo. So thank you for your attention. I'm happy to take any question.
Thank you, Will. There was so much data. It was so well presented. It was really lovely to see it. And I imagine it's just part of it. It's not everything that you guys have done. So is there any question for Will about his work from anyone? If not, Andrew, yes, please. Go ahead.
Yeah, beautiful talk, Will. Yeah, huge amount of work there. I just wondered, so if you look at the latest circulating strains and from what all the data you've got, does that allow you to predict, because kind of interesting with Omicron that it's, in the lower lung and hamsters, it's kind of lost pathogenicity a bit and gained it. I mean, what can you predict from your study, really? Can you sort of feed that into, even with AI and looking at the spike, and even actually what the sort of work Joe's doing as well?
Yes. Yeah, it's a tricky thing to address because, like I said, and we all realize that things are way more complex than what we thought. It's not just, black or white, or you change this, you get this phenotype. No, because depending on the context around, it's different. Then to predict, I don't know if I can predict. If I just try maybe to look back and understand what happened, and if we look at the emergence, the way Omicron emerged, which is very likely through some sort of chronic infection. I don't believe it's in one single person. I think it's maybe trying around in small community of maybe immunocompromised people. I think the virus kind of established itself in those people because of the chronicity of the infection, maybe in the lung or different tissues, which is maybe that has basically caused some sort of reduction in the virulence, because otherwise the host could not, sustain replication of the virus for so long. That's maybe one then Omicron emerged, and that was associated with this reduction. And then over time, by spreading it to humans, that might have regained and restore this capacity. And then if we look again at what is now circulating, it's all branching back to BA.26, which is very similar in terms of emergence to the first Omicron. So again, emerging from someone that maybe, and that, there was a chronic replication of the virus in the lungs and other tissues, and BA.26 was also highly attenuated. So now, is it just the trend gonna be like, oh, now the newer variant will basically be more capable of replicating in the lungs and maybe more virulent? I don't know. I think now, because of the very complex immunity in the population and so on, might be difficult to really predict. The only thing I can say first is I think this virus is completely adapted to human. And I think it has established itself very well, and it won't go away. I mean, I personally don't see it going away. And the seasonality, well, I'm still waiting for it, because 3 to 4 waves or mini waves now per year, it's not seasonal at all. So let's see what's going on.
Rachel, do you want to ask your question?
Yeah, I mean, it's probably more a question for the floor, but it's similar to what Andrew asked. But I mean, the big unknown in this going forward is, what is the long-term species range going to be? Because I work in the vet field, it's like, is it going to settle to being a human thing and then largely just become a human thing and not really a problem for other species? Is the range we think we've got at the moment, is it that going to be, or is that going to move? And I don't really know, but I guess with the data you're looking at and the data I've seen, and I think there's a couple of others Margaret's got in the cats, like a lot of the post-Omicron variants we're not seeing popping up in other species like we were. And it does look like it is heading towards becoming more human adapted. It's a bit speculative, and it's a watch and see, but I don't know what anyone else's thoughts are.
I mean, Dalan's lab, for example, has been doing this work in terms of, using pseudotypes for entry assay and receptor usage, whatever. And I know they are talking about having a generalist virus versus a very specific virus, and talking about SARS CoV-2 being a perfect example of generalist, which I think is completely true. Then the other thing is, I don't know. I know that we always talk about, for example, there will be an animal reservoir where the virus will evolve. We always talk about, for example, white-tailed deer. Now, I think we really should look at cats, dogs, and some other pet species, I would say, where the virus could circulate and then, acquire mutation that then will spill over back into human and then cause something. I think it is probably true, but I think it could also be that
just the other way around,
human, and then actually spill back or spill in some animal species, and actually maybe cause something in those species without necessarily changing from the human side. It's really, really difficult here. I think it's a bit unprecedented.
The big question is, where's the next one going to come from? And there's a large number of other kind of generalist candidates there that we're maybe not looking at.
You know what? If I can, especially in front of this audience, it is fine. It's not an easy mechanism, but I'm kind of a bit worried. The longer SARS-CoV-2 stays in the human population, and I think it will stay here forever, and then by being in contact with different animal species, etc., which also means in contact with other coronaviruses, like you presented. And we all know that these guys can recombine. I like sometimes, or I always hear that, yeah, but it's not like influenza. They don't reassort the same way as
Not at
often.
the same
I don't think
frequency.
this is true. It's just that we didn't look at it, and it's maybe less easy, or it's more difficult to observe it, such recombination events, compared to influenza, where basically they swapped segments. And I'm just a bit worried that then that's just going
Even
to
if it's
increase the
not common,
probability.
if you have a lot of it, that ups the chance.
And yeah, that's also why I'm a bit worried that if a new coronavirus, or let's say in that particular case, a new beta coronavirus, like a new sarbecovirus is emerging, so where even sporadically, because there is lots of SARS-2 around, you also increase the chance of, actually let's recombine because that's better for the fitness.
Are you aware
Yeah.
if I mean, we have a human coronavirus like NL6329 SARS-1. I'm sorry, I'm not aware, but do we see these cases where people are co-infected with both and then we have a recombination? Like, because I know that's something we can be worried, but I wonder if someone, now SARS-2 has been around for 4 years, 5 years, so I wonder if anyone has ever looked at it.
I personally don't
Yeah, I
know.
don't
I've
know
not seen
either.
this data.
Yeah.
I'm assuming some people are looking at it. I'm just not sure what's the prevalence of co-infection with like SARS-2 and NL63
Yeah,
or others. Maybe someone knows.
That McClaw is probably the person to ask. I mean,
oh, Lisa just wrote the Slovenian are looking and have looked at seasonal in the last couple of decades, and Hannah is also she was here before, but she also is doing that in their
there's
lab. So I
no-
guess we will just to have an idea how, like, we talk about these hypotheses, but just to have like a numerical kind of idea what we're if it's a real chance or not. So
null
I'm
data.
sorry, I think I will need to let everybody go. It was really interesting. It's really nice when people can discuss. So I just need to flag something, just a few announcements. So we still need a speaker for the April seminar. So if you know anyone that might be interested, or if you yourself are interested, you have another story you want to share with us, please just get in contact with Lisa and Jill. And then tomorrow is the deadline for the abstract submission for our annual general meeting that's going to take place in summer in Liverpool. So if you're interested, just submit and ask for a talk or poster. And finally, I was told that we have a small number of travel grants. So especially for early career researchers, if you have some master student or PhD student that would like to come, but they couldn't, we have some money for them if they are, but yeah, they need to present in order to be supported by us. So yeah, this just bear this in mind, and I want to thank the speakers for today. It was lovely presentation, and everybody for joining, and I'll see you next month.